So picking up where we left off, the video cut out pretty quickly. So back to the light reactions. What we need to remember is that the energy lost from the electrons as they travel down the electron transport chain is used in the process of chemiosmosis to make ATP. If we remember correctly, ATP has 3 phosphates bonded to it, and the way that it gets there is by using the energy from this process, because we need something to basically fuse the phosphates to the ADP. This is ATP. As we can see, there are 3 phosphate groups attached to it. And it's adenosine triphosphate, right? So we should know the different parts of ATP. The adenine, the ribose, and then the 3 phosphate groups. Why is ATP so unstable? How is ATP used in cellular work? So the things we need to remember is that those phosphate groups can readily be removed, making it an unstable molecule. Also because it is charged, those phosphate groups have a negative charge. ATP is used to do cellular work because when we break those bonds, we release a large amount of energy. So what we need to know is that the protons, the hydrogens that are sent out of the photosystem, are pumped from the stroma into the thylakoid space, which is across from the thylakoid membrane. This builds up a concentration of hydrogens in the thylakoid space. Think about the way that we know that the nervous system works. A similar thing is happening. Basically, we create a concentration gradient, and this creates potential energy. So it's not energy that's being used yet, but it has potential energy. And chemiosmosis, the protons basically build up so much that they diffuse back into the stroma using ATP synthase, and ATP synthase is then powered to turn ADP into ATP. This is known as a proton motive force. Essentially, the protons shoot through that protein, and the synthase starts to spin and create ATP. Basically like a turbine forcing ADP and the phosphate group to bond, and that forms ATP. Again, so this is what it looks like. So we've got the thylakoid space, which is inside of the membrane. We've got my thylakoid membrane, and then we've got my stroma, which is the fluid outside. Essentially what happens is, let's see. All right, so our energies from our photosystems leave and they move into the stroma. They are excited. The next thing that's going to happen is the electron from photosystem 2 moves to photosystem 1, allowing hydrogens to come into the thylakoid. Then the electron from photosystem 2 is going to move and create NADPH and also allow more hydrogens in. We can now see that we've created a concentration gradient. All of those hydrogens start to leave. They make ATP synthase spin, and it creates ATP. Also during that time, water is split so that we can have my electrons back in photosystem 2. Remember, photosystem 1 gets its new electron from photosystem 2. The rest of those hydrogens go back to creating that gradient, and the oxygen is lost into the atmosphere. This is another module, but we are not going to look at it. This one's a little bit more complicated. Feel free to look up the McGraw-Hill animation if you want to see this. Again, so a quick recap. In the light reaction, the energy is light. It is used to excite electrons to make ATP. It also produces NADPH, and those 2 energy sources are going to power the Calvin cycle, which is the second step. The light reaction occurs in/across the thylakoid membrane, but it's all inside of the chloroplasts, which are inside of the cells, inside of the leaves. Light and water are required. We know the light is what gives the energy to the electrons to leave those 2 photosystems, and the water is going to be split up to provide new electrons for the photosystem 2, which has lost its electrons to photosystem 1. Oxygen, ATP, and NADPH are produced during this cycle. We see that there's this cyclic electron flow. Basically, the electrons start in photosystems 2 and 1. Photosystem 2 gives its electrons to photosystem 1. Photosystem 1 gives its electrons to NADPH, and then water is split to provide electrons for photosystem 2. Cyclic phosphorylation is a very ancient biochemical pathway. Most photosynthetic bacteria and all photosynthetic eukaryotes use cyclic phosphorylation. Because cyclic electron flows produce ATP, they don't produce NADPH. And so I misspoke above. We know that our photosynthesis that we are looking at is not cyclic because those electrons that leave with NADPH do not re-enter the cycle, whereas in a cyclic electron flow, the electrons stay within the cycle all the time. Only photosystem 1 is used, electrons are recycled, and water is not split. So this is in bacteria and photosynthetic eukaryotes, so not plants. This is what the cyclic pathway would look like if we were curious. We don't really need to know this for the AP test, though. So this is our image again. One more time, we see the photosystem 1 sending the electron out, and this is showing the cyclic nature of photosystem 1. When we add in photosystem 2, we don't have a cyclic pathway. So in this case, we are just making ATP, and then NADPH loses its electron again back to photosystem 1, so it is cyclical. Again, this is not in plants. In plants, we have photosystems 2 and 1. This is just a kind of synthesis of the two types of electron flows. What is most important is that we understand the non-cyclic. If you can understand the non-cyclic electron flow, we can understand the cyclic electron flow. Okay. Putting all this information together. The light reaction uses the energy in sunlight, and it makes it into chemical energy, specifically ATP and NADPH, which are going to power the Calvin cycle. Light and water are required for these light reactions, unless it is cyclic, right? And then it doesn't need water. ATP, NADPH, and oxygen are produced unless it is cyclic, and then only ATP is made. And again, those red ones are denoted if they are not produced during cyclic phosphorylation. There are some kind of experiments and videos that you can look up if you want to. It is not necessary though. And again, this animation was at the beginning of the video. Feel free to look at it if you would like to.